So, you know, we were looking around and looking around. I finally broke down and bought a microscope that cost $2,000. And then we found this one. And this was actually better than the $2,000 one that we bought. The $2,000 one that I got, that lamp gets really, really hot, and you gotta always worry about burning up your table beneath your microscope. Great. So I would very much recommend this one. So it's about $300 without the camera. Well, actually it's about $250. Depends on what the exchange rate of Canada actually is. And then $350 with the camera, $300 to $350. Making certain that you get a microscope with an ABE condenser, ABE condenser. It also has an iris and diaphragm. That's extra, okay, is with iris and diaphragm. That's a separate word. Yeah, made it squeal. It's a Trinoc, and then the ordering number is M8311. And there's the website, microscopenet.com. So $279. Ask for Raul. Here's his phone number. And he can switch out the 100x objective. If we don't need it, we don't have to use that one because we don't need that high magnification. We don't dry our organisms, so you don't need the high mag. And I think you might want to play around with really high mag, just leave the microscope the way it is.
You don't need what?
You don't need the 100x objective
okay.
for what we're doing. So you could switch these two things. On the other hand, some people, they think they might want to be looking at things and need that high magnification, so you could leave it. Up to you to make that decision. There is a metal travel case, so if you plan on traveling a lot, we're going to So there's the ordered information for the metal travel case, in case you want that. If you're just driving around the island, no big deal. These little microscopes can just go on the back seat with maybe a pillow or something around it. And then the microscope panel is inside. Although when you call and talk to Raul, you might ask them if there's a better one on the market. So there's the ordering information for that digital microscope. It comes with the software that you need. So if you call and talk to Raul, ask him if he can include the camera with the microscope, so you don't have to order it separately. Up to you.
Is the first microscope, it's not digital, right?
The microscope doesn't have any connection to laptop worthy things. So the microscope is just the microscope. This thing is just a microscope.
And it doesn't need to be plugged in, or does it?
It has a lamp, so you're going to have to plug it in to some electricity in order to get the lamp to work.
Yeah.
And so,
Could you put a mirror underneath or anything?
you cannot get a bright enough amount of light if you've got a mirror. This isn't going to work for the kind of samples that we're dealing with.
Okay.
You really need one a lamp. That's what you want. I think it's an LED lamp on this microscope, so it lasts forever. But you can get a connection that you can hook it up to your car battery, so that's possible. So you can get something that also goes in your cigarette lighter.
Well, LED is, I mean, that'll be fine. I just have a solar array, like a small solar array, so LED is low one or whatever.
It doesn't take much electricity,
Yeah.
and you get some pretty good bright light on it. So the person to talk to will be Raul, and see what he has to say, and ask him to send the camera along with. Okay.
I've got another question. The India ink. I've got a friend picking some up tomorrow. Is one bottle enough? Yeah.
Okay, should be. Should be more than that. Here I go on it. I'm gonna turn this off.
I'm going to go to
on the floor. And the rest of this conversation for today is covered in this ecological monograph. So Ingham et al. 1986. That's my husband who's the first author, I'm the third. So the data are in this paper. If I presented all the little graphics and the tables of data and exactly went through the whole experiment, I could put you to sleep so fast it would not be funny. So I hope you'll let me go ahead and present it in a more interesting fashion. And in less scientific ease. When I need to go to sleep at night, this is the paper I go get. Because it is just, you read a paragraph and you go, oh, what did that mean? And you go back and read it because it's just so dense in information. So, great way to go to sleep at night. So, starting at the beginning, photosynthesis. Here's where energy is made in your plant. So I want everybody in the room to get a mental picture in your mind of your absolute most favorite plant on this planet. Mental image, think of what it looks like. This happens to be my favorite plant. It's a baobab tree, and it's in South Africa. I also like baobabs in Northern Territory in Australia, so I don't really care where they are. But one of my favorite trees in the whole planet. You just have an aura of energy about them that's so amazing. This tree was here before the farm went in, so it was here before the tomato and the whole problem with rotations. Sitting out in the middle of an agricultural field, and they have left this baobab tree here. I am standing in this picture. I don't know if you can quite see me, but that's me in the inside of that tree. All of the heartwood has been hollowed out by the termites, but the phloem and the xylem and the living part of the tree is still there, still intact. So my favorite tree. So we've all got your mental picture of your favorite plant in your mind. Now let's be honest with me, how many of you actually visualized the roots on the bottom half of the tree? If we forgot as much of you as you just forgot of your plant, would you be alive? No. What have we been talking about all morning? And you've just been trained your whole entire life to give no consideration to what you don't see. And yet this plant is like an iceberg. Most of this plant is below ground. So especially when we look at perennials, more than half of the tree is down below the soil. And if we forget more than half of you, you'd be so dead. So whatever half you want to leave behind, that's what you just did to your plant. Now, there's some variation when we look at roots. How much of the energy, so if we go above ground, 100% of the energy of that plant is fixed as photosynthetic. Those sugars produced by photosynthesis, some of them move down into the root system and stay there. How much of that 100% energy of the plant moves down into the root system? So here's a fun one when it comes to weeds. Weeds only put 20% of their energy into the root system. That's all that goes below ground. So see where weeds aren't really going to be benefiting you much? They don't change the soil very rapidly, but given enough time they will. And we move on to the next stage of succession. When we're looking at grasses, when we're looking at veggies, when we're looking at that next stage of succession, 60% of the energy of those plants goes below ground. When we deal with vegetables that have a tuber, so when we're looking at potatoes, when we're looking at onions or garlic, 75% of their energy goes down into their root system. When you're looking at tulips, when you're looking at taro, 75% of the energy of that plant is now down in the root system of the plant. If we forgot 75% of you, there's not much left to be functioning, is there? When we're looking at perennial plants like shrubs, deciduous trees, conifers, 80 to 85% of the energy goes down into the root system and stays there. So from now on, when you look at the above ground part of the plant, what are you going to be remembering to also think about? how much of that plant's below ground, and how important that is to the function of that plant. What does that energy do as it goes down into the root system? What does it get used for by that plant? Well, part of that energy is going to build structural roots. What do you need structural roots for? Well, your tree's taking your branches up into the sky. You better have a root system that's going to help balance you and keep you upright when the wind blows. When you're on a slope and water is rushing down that hill, you better have a nice, stable, deep root system, or you're going to be joining that water going down the hill. So we've got to anchor our plants into the soil, and so a major function of building these root systems is to keep it in place so it doesn't blow away and it doesn't blow over. When you have a crop that falls over in the wind, it's because your root system probably was not going down deep enough to keep that plant upright. There wasn't enough support. And so over it goes, and you're not going to be making any crop. You're not going to be getting any money from that crop. So part of the energy also is going to go into building lateral roots. So lateral roots, and those lateral roots take up nutrients by simple diffusion. And we talked about the chemical paradigm, about how those nutrients are taken up. And hopefully you now realize that that's not quite right. You've got to go on and understand what's really going on. If you've got a whole bunch of bacteria and fungi, protozoa, nematodes, microarthropods in your soil, how does nutrient cycling work? So yeah, we take up nutrients into the root by diffusion, but what the plant actually obtains is just those soluble nutrients that are right here, right next to the plant. They don't come from very far away and move into the root. So we need to change our thought patterns about what's actually going on inside of root systems. 50% of the energy going down into your roots is going to be released into your soil as exudates. We've talked a lot about these this morning, we've talked the last couple of days. Exudates, 50% of the energy going into the root systems comes out into the soil in the form of exudates. So when we're looking at trees, if 80% of the energy, 85% of the energy from above ground is going down into the root system, and half of that is dumped out into the soil, that's 40% of the energy budget of that plant is being pumped out into the soil. Why are we doing that? So what is an exudate exactly? What is that stuff? Exudates are made up of simple sugars, mostly simple sugars, a little bit of protein, and a little bit of carbohydrate. If I sent you into a kitchen and asked you to make a recipe of mostly simple sugars, proteins, and carbohydrates, what am I asking you to make? Cakes and cookies. So, bowl. What we're going to put simple sugars in here. What are simple sugars in your kitchen? White sugar, brown sugar, honey, molasses, syrup. So think of all the great types of sugars that could be going in there. So what exactly we're going to end up? It depends on which sugar that you choose. What combination of sugars? Protein. What's protein in your kitchen? Throw some eggs in there. Throw some milk. Okay, so just stir that up. Let's throw in carbohydrate. What's carbohydrate in your kitchen? Flour. So what is this a recipe for? Cakes and cookies. The plant is going to be putting out foods to grow the microorganisms around those root systems. Does the plant know that if it puts out this kind of cake, it's going to grow these bacteria? If it puts out that kind of cookie, it's going to grow these fungi? Absolutely, it knows this. For the whole entire existence of plants on this planet, they've been doing this. For the last billion years, they've been working on this interaction. Maybe they've had a little bit of time to figure this out better than you and I understand it. Because we have paid no attention to this interaction at all. So from now on, when you get that mental picture in your mind of your plant, not only are you going to remember the root system on the bottom of that plant, right? You remembering that? I want to see everybody shaking their head yes. Okay, yes. And now what else do you have to picture coming out of the root system. All those cakes and cookies. Because over here comes the lemon meringue pie. Because over here, this plant requires some more boron. And if it puts out lemon meringue pie, it's going to grow the bacteria or fungi that can solubilize boron straight off the sand, the silt, the clay, the rocks, out of the organic matter. So we're getting that boron into the bacteria and fungi. Over here, Phytophthora is attacking my root system, and we better get some Christmas cookies out there in order to grow those bacteria and fungi that are compete with, consume, inhibit those disease-causing fungi. Over here, your plant needs a little bit more iron, and so it's putting out birthday cake in order to grow those organisms that will pull the iron and put it into bacteria and fungi so it can be cycled into a plant-available form of nitrogen. Plant-available form of iron. So all of these different food sources. So how many species of bacteria and fungi do we have to have in that root zone? So that no matter what the plant requires, the plant can get precisely what it wants as rapidly as it wants.
Can we assume that the candles are beeswax?
Could be, yeah. Not the synthetic waxy stuff. Not paraffin. Maybe. I don't know what bacteria and fungi are promoted if we put paraffin into the soil. And there are bacteria and fungi that can use paraffin. So different kinds of foods going into the soil. So think about this. Over here, this is bacterial food. But over here, this is fungal food. So what's the pH over here? What's the pH over there? It's not at all the same, is it? Over here, bacterial, fungal, bacteria, fungal. So the pH could be 5.5, 6.8, 12, 11, 7. When you do a soil pH, what are you actually learning about? Because we take a soil core from here, and from over there, and from over there, and over there, and we mix it all together. Where did you take those samples from? And what's the mix of biology in those soils? We take that soil, we mix it all up, and we're going to take 500 grams of that, throw it into a beaker. We're going to add 400 mils of water, stir it up, and then stick our pH probe in there. So what are you finding out about? A sum of all of the pH concentrations in all of that soil from all of those different places. So it's an average. What does that tell you about whether your pH was 7, or 8, or 12, or 5.5? Fairly meaningless, isn't it? It's an average. So maybe you really want to be focusing on just particular parts of your soil. You really want to be focused on taking soils from very specific places so you find out what's going on. Fungal, bacterial dominance. Or maybe it's just easier to look at your fungi and your bacteria in your soil so you can figure this out. Now, so we got all these great cakes and cookies growing billions and billions of bacteria and fungi all around, all over the surface of that root. Billions and billions. Your best Carl Sagan imitation. Billions and billions of bacteria, miles and miles, or I guess Carl would say kilometers and kilometers of fungi, protecting that root system. All of these bacteria and fungi are capable of competing with, inhibiting, and consuming the disease-causing fungi, the disease-causing bacteria, preventing the pests from even knowing your roots are there. So we are talking disease protection here as well. When we're starting to look at disease protection, we want to make certain that all the infection sites on our root systems are already occupied by somebody beneficial. On your roots, there's a cute little root, maybe even a root hair coming out there. We have specific lecithins, and those are the only sites that our pathogens can interact with, and then allows that pathogen to find a pathway through the cell wall and the cell membrane of that plant and start to cause disease. So if it's very specific lecithins, isn't that another cute word? A specific type of protein carbohydrate mix, that lecithin has to be present. Pythium cannot attack your root system unless it has the opposite to that lecithin. If perchance it can find that lock and key, now we're going to have disease, now we're going to kill the plant. Only if Rhizoctonia can match that lecithin, now we can have disease. So what we want to make sure, with all this great diversity of bacteria and fungi here, is that we're going to grow those bacteria and fungi that get to that lock and key mechanism and occupy this infection site long before any disease can possibly get there. So a number of different mechanisms. We're occupying all the space around that root system with our billions and billions of bacteria, our miles and miles of fungal hyphae. We're making sure any exudate coming out of that root system is eaten by a good guy, not a bad guy. We're making sure all the infection sites are already occupied. And then, perchance, if a disease-causing fungus does germinate and start to grow, we want to make sure that we have predators of those fungi that are happily chowing down maybe our fungal-feeding nematodes. We want to make sure we have those cute little microarthropods that are going to attack and consume. These disease-causing fungi can never make it to the root system. We will never have disease. There is absolutely no need for those pesticides. How about above ground? Are there cakes and cookies that come out of your plant material all over the plant? Every part of the plant is releasing cakes and cookies. So really, this picture is not right. What's wrong with this picture?
No
Cakes and cookies above ground. So we better from now on imagine cakes and cookies coming out of the above ground part of the plant. Again, just as much specificity. We have to protect every part of the above ground from all the diseases and insect pests, and the plant does that by making sure we have the right kinds of bacteria, fungi, protozoa, nematodes growing on all of the foliage, on all of the branches, on all your petioles, on all your flowers, on all your fruit. You need to have them covered and protected. Same mechanisms. Now notice that above ground, the cakes and cookies are mostly completely different. Where did the inoculum of microorganisms on the leaf and branches and everything above ground, where did that original inoculum come from?
Blown in on the wind.
Where did it originally come from?
The ground.
Because remember the seed? Seed germinates. Here goes the first true leaves, completely covered with soil organisms. So first true leaf stage, now we grow some more leaf materials, so all those cute little protozoa, the microarthropods crawling up the stem of our plant are bringing along taxicabing all of our soil organisms and spreading them all over. But now these are different foods. So the foods up here selecting for different organisms, the ones that are particular to above ground. So the biology of above ground may look very, very different in terms of species and number of each species, but these are all soil organisms. They are no different from what's in the soil. It's just down here we have different foods, up there we have different foods, so it looks different. Don't make the mistake to imagine that we have a completely unique, not found anywhere else on the planet, set of organisms above ground. Think about a fly in the soil. And then it takes off and it lands on your leaf and it walks around in the leaf and it walks over here to your fruit and it walks up the stem. What's it leaving behind? It's a taxicab. It's leaving behind all those organisms it encountered when it took a little landing on your soil. Think about a bird walking around on the ground and then it lands on your plant. We're leaving behind that inoculum. Birds walking around under a plant plant surface, and then it hops down to the ground. It's going constant communication between above and below ground. Purpose: prevent disease, prevent insect pest attack. So we just want to make sure we get all of those surfaces covered. How easy is that? So, would any of these foods feed pathogens? What would happen if your plant put out a cake or a cookie that encouraged the disease-causing organisms in the soil or on its surface, a foliar surface? Well, then you'd have disease, wouldn't you? And now you've got a dead plant. End of the story as far as evolution is concerned. If that plant dies, no plant's stupid enough to do that again, is it? So think of the years of selection, where any plant that put out a food that encouraged the disease-causing organisms, it died. And so, evolutionarily speaking, none of those plants survived. So of course, the cakes and cookies going out into the soil or your leaf surfaces above ground are only going to be growing good guy organisms. So, disease suppression mechanisms, there you go. You got them all listed right there. We're not going to have any food for somebody that's a pathogen. We produce inhibitory compounds. You know, people always think of massive amounts of antibiotics being produced. But when you look at organisms in the soil, how far from the organism does an antibiotic usually get produced, especially when you're in a soil situation? It's not very far from that particular organism. We don't have massive amounts of antibiotics being produced in the soil the way we do in our pharmaceutical industry. When you take a pill, an antibiotic pill, it is a massive concentration of that antibiotic, and it affects your whole body. That is not how it operates in the soil. Very site-specific, very local. That organism is only affecting the area around its body, not a massive part of the root system. So, is it really that important a mechanism? Maybe. Maybe not. Got some work. So, here's your charge to some of you. You got to work on this one. Lots left to do to understand what's going on in the soil. So, why do disease organisms attack plants? Because you didn't have the right biology around that root system, around that leaf, to protect your plant. Stressed plants can't make the exudates. They can't make a solid cell wall. They're lacking calcium, they can't protect themselves. And if you get a huge number of disease-causing organisms, you can overcome the defenses of anything. As I cough and sneeze and blow my nose. So most diseases require high nitrate levels and low oxygen. So when we start going anaerobic, when we have masses of bacterial growth and then masses of protozoa coming along after releasing that nitrate, that's where we get problems. So, going back now to nutrient uptake. This is not how it works in soil. The nutrients, the soluble nutrients moving to the root system are taken up by those bacteria and fungi. Growing in there billions and billions and miles, there's no way these soluble nutrients can get into your plant. Who's feeding your bacteria and fungi? Plant. So golly, how can this work? So we've got to understand a little bit more about all of this. I want to go through just a little bit the organism numbers in soil, what's typical. So we're looking with biomass, total bacteria across the top, biomass, micrograms biomass per gram per weight of soil. The next one, number of species. And then total fungal biomass, number of fungal species, and then mycorrhizal colonization. This is an endomycorrhizal, not an ecto, because now we're talking about most of our ag systems. So in our typical ag soils today, we have 400 micrograms of bacterial elements. We have 5,000 species. See a problem there? There's only 5,000 species. How many do we need? 75. So right there, we don't have the organisms in our typical ag soils. Where'd they go? How come they're not there? We killed them. And they have not yet come back into our soils. I have to kind of giggle when some soils guy says, yeah, sure, we destroyed all that life in the soil, but they just come back. The next day, they just come back. Really? You just nuked your soil. You just did Hiroshima. How fast did human beings come back to Hiroshima the next day? How long did it take us to move back into a nuked area? It can take a very long time. Now, let's say you have a clear cut that's only about 200 feet across. Well, your inoculum's right there, about 200 feet away, so it can move in pretty quickly. What if you've nuked a 5-mile square area? How fast for that biology to move all the way back into that middle of the 5 miles? We can show you places in the Pacific Northwest where we have clear-cut patches where the beneficial biology has not yet, 150 years later, we still do not have a complete food web back into the middle of that 5-mile square clear cut. What if we clear cut most of the Great Plains of the United States, which is sort of what we've done, isn't it? How long is it going to take that beneficial biology to move hundreds of miles? We destroyed that biology, we have to put it back. As global climate change happens, and the habitat for all these organisms move northward, they are not keeping up with that changing climate. They're not moving north fast enough. Who's going to have to take care of that problem? Us. We caused the problem, we better take care of it. So do we have some work to do? Mm-hmm. You gotta get that biology back into the soil. This work that I'm talking about here, this is done with DNA analysis, so all of this DNA analysis, and this work on the ag soil was done by Joe Teaching at the Center for Microbial Ecology at Michigan State University. Looking at fungi, 5 micrograms. What do we need here? At least 300. So look at how much isn't there. We've lost most of that function in that soil. We have 500 species of fungi. This is work from Tom Bruce at the University of California, Berkeley. How many species do we need? 25,000. So we are lacking protection. No mycorrhizal colonization on the root systems of plants that require mycorrhizal colonization in order to grow without pesticides or inorganic fertilizers. No wonder we're in trouble. Now, around the rhizosphere, where your plant is putting all kinds of cakes and cookies in there. Yeah, we're going to grow a lot of bacteria, but we're lacking species. So there are many, many days where your plant is unprotected against disease. Our fungi, yeah, we're putting out fungal foods, but we just don't have any species of fungi to grow. We do not have adequate level. We have not improved that diversity. Still no mycorrhizal colonization. We have plants that are going to get sick. Healthy soil. 350 micrograms of fungal biomass, 25,000 species. Now this work was done by one of my graduate students at Oregon State University. And it was way back in the early stages of trying to figure out how to do this species diversity thing using DNA analysis. So she did the best she could with extracting just the bacterial DNA or extracting just the fungal DNA. And so using different primers to pull out just bacterial, just fungal, and then sequencing every single sequence of bacterial or fungal DNA. So she had at least 40,000 strands of DNA to go through and look at this sequence and then compare all of that data. That's a full master's degree in order to do that kind of analysis. So we got done and we tried to publish this table of data. And what the scientific community said was, you are crazy, lady. You cannot possibly even get 25,000 species of bacteria in the soil. There's no space, there's no room for them. It's not possible that you can get 8,000 species of fungi in the soil. And I was just like, where's your data to say that? What are you people stupid? So we struggled with it, we tried to get them to understand, could not get it published. Today when we try to get this published, now what's the criticism? You have underestimated. You don't know what you're doing because this should be 75,000. You don't know what you're doing because this should be 25,000. We were just too much ahead of everybody else in doing this kind of work. So, oh well, you win some and you lose some. And this is one of those, no matter what I do, I'm going to get people criticizing me. It happens. So, looking at that healthy soil, you might want to change this number in your notes to 75,000. Work done at Auburn, work done at Duke, work done at Cornell is saying very clearly 75,000, 25,000. But now in a healthy soil, look at the mycorrhizal colonization. We have biology needed to protect those root systems. Now around the root system, healthy rhizosphere, we're growing lots of different species, protecting that root. With the bacteria, lots of different species, massive biomass of the beneficial fungi around that root system, protecting that root system, and we have 55% colonization of that root system with mycorrhizal fungi. There is a paper by Brent Reeves from Colorado State University that shows that you get benefit. So you start getting benefit from endomycorrhizal colonization when your roots are at least 12% colonized. 12%, you start having benefits. However, you don't get all the benefits until your mycorrhizal colonization reaches 40% colonization. We don't have to have it above 40%, but it's probably safer to have it up a little bit higher. So this is satisfying the demand of that plant for the mycorrhizal colonization that it requires.
What type of data do you get from like Soil Food Web Lab? Is it anything like this? Or?
Yep. So when you would get from the Soil Food Web Lab would be this total bacteria, you would get total fungi. They can give you an estimate of diversity of both of these, and they will give you active bacteria and active fungi. They'll also give you protozoa, flagellates, and uniciliate, so you can separate the good guys from the bad guys. And they will give you all 4 kinds of nematodes: bacteria feeders, fungal feeders, predatory, and root-feeding nematodes. And they will separate those out for you to the genus level, so you know exactly what you have. So, well worth the money you spend for it. Now at Rotale, we give you general values. We're giving you the qualitative information that we're going to teach you how to do on Friday. Quicker, easier, qualitative. So if you really need quantitative data, you need to send it to the lab in Oregon or New York or England or New Zealand or Australia or there's a couple other places, whatever. Okay? So, coverage and castle walls, I think we've pretty well covered that. Nutrient retention. Bacteria and fungi are the things that retain nutrients. They require so much nitrogen, phosphorus, sulfur, magnesium, calcium, potassium, sodium, everything else. And those nutrients stay inside their bodies and they glue themselves. They bind themselves. They don't leach. They're not going to wash away. So think of bacteria. It's not going to be just sitting in the soil solution going la-di-da. It's got to be glued to its source of food. Because imagine what that little bacterium would say if the water comes along and starts washing it, like, wait, that's my food, no! So think of these other little voices in soil screaming. No, they don't do that. They're glued. They're bound. So they're not going to wash away. Here's a picture. Leaf surface from a corn. This was picked off the plant. Then it's a scanning electron microscope shot. So we're looking at something that we have critically point dried. So these organisms have shrunk down a little bit when you remove the water. And then we're going to coat this with gold palladium. There's about 1 to 2 nanometers of gold palladium coating all of the surfaces. And now we hit this with a bunch of electrons, and we can take a picture of how those electrons are reflected back. And so what we see is all that surface phenomenon. So corn leaf, what all are you looking at here? See all this oozy, slimy stuff on here? What is that? Those are the glues that the bacteria make. All of that is glue produced by bacteria, copious amounts of slime. And these are the individual bacteria. Right there, there's a colony of bacteria, and there's another. So just go around and find all the little dotty things. Those are bacteria. Some are rock-shaped, some are round, coccyx-shaped. So all of that material, bacteria down and meshed in that glue layer. What are all the strands? What are all the threads on that surface? Fungi. Look at all the crystalline material on the surface of those fungi. That's oxalate. Oxalate, or another way of writing that is oxalic acid. Same thing, just a different way to express that same thing. Oxalic acid is produced by the fungus, and they are putting those crystals, and notice we've got all these little crystals on here, we've got all those little crystals on, not all fungi are making oxalic acid, and well over 50% are producing oxalic acid in order to instantly grab any positively or negatively charged ion out of solution. Immediately, instantaneously. So the fungus, when it needs whatever got grabbed, when it needs it, it just reaches outside and grabs it, pulls it in from the pantry. So just think of this as a fungal pantry sitting out here. So they're collecting nutrients whenever they want it. It's available right then and there. So all these strands, all these threads. Take a look at the diameter of that fungal hypha. How can you tell what diameter this is?
The bacteria.
Yeah, look at the bacteria. This is 1 micrometer. So how many of your little bacteria would fit across there? So we're looking like 10 micrometers across. That is a beneficial fungus, beyond doubt. Now some of the fungi are narrower diameter. Some are a lot narrower. We've got actinobacteria. The diameter is the same size as the other bacteria. So morphology, we can tell what we're dealing with in this sample. Is this bacterial dominated or is it fungal dominated? Kind of hard to guess. Look at all those fungi, look at all those bacteria. And we're going to have bacteria down in the slime land. How many protozoa in this picture? There's a whole bunch in here. Can I find the protozoan? I know there's one up on the top here. We got a couple of nematodes in this picture, but they're down in the slime layer. So imagine a bacterial-feeding nematode in this goo. It's like having Thanksgiving dinner. Just having a grand old time chowing down and making babies really rapidly. How many of you can find bare leaf surface on this picture? I'll give you $1,000 if you can point out bare leaf surface on this thing. If you're wrong, you owe me $1,000, so come on, be stupid. There is no bare leaf surface. Next time you pick up a leaf of a plant, think about what you're actually touching. You know, a lot of guys, people go, I'm touching bacteria. Oh yeah, guess what? What did I just touch there?
Bacteria.
What did I just touch there? Yeah, we're all protected by layers of bacteria, fungi, protozoa, maybe even some nematodes. Anytime you grab anything, you're not actually touching this object. You're touching the bacteria living on the surfaces. So don't get weirded out by that.
How old is this leaf? Picked fresh off the plant? Or
Yeah, it's fresh off the plant.
So this is, all this is existing just even as the plant is growing?
Yep.
Okay.
Now, what would this look like if this corn plant had just been sprayed with pesticide?
How would you see some leaf surface, right?
A lot. Almost all of this would be gone. So how toxic is your pesticide to these beneficial organisms living on the leaf surface? Do some before and after scanning. Kind of fun. A little spendy. That's cool. Palladium, guys.
What magnification size?
This would be, well,
Roughly.
we're probably looking at 1800 totally. So it's not real high magnification, especially for a scanning electron microscope. But we're trying to see surface phenomena, and that's what scanning microscopes are really good for. So, going over, bacteria make alkaline pH material, fungi make acidic, so between 5.5 and 7. There's in the not too recent past, there was a person from the USDA who said the only glues in soil are produced by fungi, and they're called guanulin. And I read that paper and I went, what? Reviewer, let this one by, because that's absolutely ridiculous. Fungi don't make much glue. How do you know? Well, take a look at that one. Look at bacteria, just a mesh in the glue that they make. The fungi, there's glue on there, but not a lot. So, fungi make the only glue in soil? Don't think so. So, every once in a while, still in scientific literature, weird stuff comes out. A really great book to look at some of these slime layers for the bacteria, how much the fungi, what do these organisms look like around root systems, is this book by Foster called The Ultrastructure of the Rhizosphere. Just fantastic pictures of these critters as they're growing and living around root systems. Have some fun with that one. So what is the concentration of nutrients in a bacterium or in a fungus? Who wins in the competition for who can grab nitrogen faster? Who can grab phosphorus or calcium or sodium or potassium? Who wins? Bacteria, fungi. Question?
Oh, well, we were talking about the pH, and you were talking about how the bacteria is typically like greater than 7, and you got your fungal that's between 5.5 and 7. And if there's this right balance between them, then shouldn't that make the pH like sort of near neutral? It's not that 11 that alfalfa field was?
Yep. So, that alfalfa field. When we're looking at alfalfa, what is the fungal bacterial ratio that alfalfa requires? So it's non-bacterial. And the actual species of bacteria we had in that alfalfa field made glues that are way up there. Seems to make alfalfa really happy to be up there. Didn't make much difference. Now, also, when we went into the root system of the alfalfa, go to the root system, not just the soil. So, see, I tricked you. I talked about the soil pH. But now go look at the pH of that alfalfa in the root system. pH in that alfalfa in that root system was 7. So you gotta look out where you're taking your pH, because you can kind of fool yourself about what's going on.
What about the leaf tissue sap pH?
You didn't look at that. I haven't looked at that.
Because, like Britannia thinks, well, thought that it should be like 6.4, and that if you vary off that, then you've got to do too much like bacteria, too much fungus.
Yeah, I've never really looked at that. What versus, in the sap in the leaf tissue.
Yep.
What's the pH that it's supposed to be?
Yeah.
I don't know. Should I care? Probably. There's something more I need to learn. So, let's look at the nutrients held in bacteria, protozoa, well, people, green leaves. So we're going to go through this whole list. Bacteria are the most concentrated in any nutrient you want to talk about. No other organism on this planet has more nitrogen, more phosphorus, sulfur, magnesium, calcium, sodium, potassium, everything else is highly concentrated in the bacterial biomass. They win hands down. Head-to-head competition with anything, bacteria are going to win. Their enzymes beat anything on this planet. If you had to go head-to-head with E. coli, who would win for that nitrogen? E. coli's got it beat. There is not, you wouldn't even know. They're that fast. But bacteria can only breach what's right around them. So if that nitrogen is way over there, and that phosphorus is way over there, and that sulfur is way over there, bacterium can't do anything about it. It's not that mobile, it doesn't move that far, so it's got to wait in place for everything to come by. So as long as we don't till the soil, that bacteria may not get to be in a place where all the nutrients are right here. So every time we till, and we mix all of those nutrients together, then the bacteria is going, "Weehaw! I'm going to win." Whereas fungi, they grow as nice long strands. So that hypha can go over here and get the nitrogen, that branch can go over there and get the phosphorus, this end of the fungus can go get the calcium and the potassium and the sodium, and it pulls it all together. So when our nutrients are spatially spread out, fungi win. Both of them hold nutrients. Which one holds more? So bacteria, of the carbon and nitrogen ratio, 5 to 1. And we look at phosphate, the calcium phosphate, calcium to sulfate, calcium to anything else. Similar kind of ranking in most of these organisms. Bacteria hold more nutrients than anything on the planet. Fungi come in second. Now, just to make it a little bit more complex for you, fungi grow as long strands. And the tip cell is where the growth is occurring. So in the tip cell, the cell wall is very narrow. There's not a lot of cell wall. It's not very thick, not holding a lot of carbon in that tip cell. And that tip cell is completely cytoplasm filled. So the tip cell of the fungus has exactly the same carbon and nitrogen ratio as bacteria. That tip cell's grabbing stuff. Bacteria have better enzymes, but fungi come in pretty close. Head-to-head competition, the bacterium is going to win. Fungi is going to win when the nutrients are spread apart. As we move back from that tip cell, you may have to actually go a couple of cells, so I'm going to collapse this down a little bit. That next cell may have some great big vacuoles in it. And those vacuoles are where that fungus is putting away waste material. It doesn't need that waste, and it's going to out into the soil solution, but it is empty. So it's not as cytoplasm filled. And it puts down 2 layers of cell wall. So now when we look at the carbon and nitrogen ratio of that cell, we're looking at a ratio of about 20 to 1. Keep moving back along that fungal hypha. Sooner or later we're going to get to cells that have almost no cytoplasm, very little enzymatic ability, not very active. It's pretty much going to sleep. And it may have 3 or 4 layers of cell wall laid down. Notice it lays the cell wall down inside, not on the outside. The outside does not get any wider. It's laying that carbon on the inside. So the only part of that cell that's C to N ratio 5 to 1 is just that little bit here. So this cell has a carbon to nitrogen ratio of maybe up around 100 to 1. Keep going, you're going to have absolutely empty cells where a really thick cell wall has been laid down. So can you see why fungi are very conserving of the carbon that they're using? They're leaving behind a carbon pipe, and it stays in the soil. So as you keep going, these cells may have a carbon and nitrogen ratio of 300 to 1. So what's the average? What is the C-to-N ratio of fungi? Well, it depends. Very good. How rapidly is your fungus growing? Let's say your fungus is going this way and it can't find any food, and it can't find any food, and it can't find any food, and it finally says, you know, it's just not working. I'm not finding any food. So that tip cell, that cytoplasm turns around and comes back this way. And somewhere along the line, then it's going to start growing a branch, and it's going to start growing out this way. Looking for some more food, looking for some more food. Don't find any food, it's going to turn around and come back, and until it finds some food, it doesn't start branching. It doesn't really start growing really rapidly. So C-to-N ratio on a fungus. If you were a fungal feeding nematode, what part of this fungal hyphae are you going to eat? The tip cells. You can go right down there because it's got something to suck out. If you're a microarthropod, what part of this fungus are you going to eat? Ooh, trick question. Because you have to know what else the microarthropod has been eating. If a microarthropod is in soil that has a lot of humic acid, that has a lot of very recalcitrant carbon, it's going to come and eat the empty cells, because this is like Pepto-Bismol. Microarthropod's got an upset stomach from all of that humus, from all that complex recalcitrant material, and it's got to neutralize it with empty cell. And other times, it wants the tip cells too. So lots of interactions to understand. So bacteria and fungi, they come in first. They're concentrating more nutrients than anybody else. What's your carbon to nitrogen ratio? Let's see. See how bacteria and fungi have to beat? We don't even come close. Because we got lots of bones. We've got connective tissue. We've got all kinds of wide C-to-N ratio materials in our body, so our carbon to nitrogen ratio is wider. We're still pretty juicy organisms. What's the C-to-N ratio of meat? Yes, your muscles, those are meat. It's a shock for a lot of people to think about that. C-to-N ratio of your muscle tissue, 10:1. So parts of us are way more concentrated in nutrients, other parts are not. But on average, in general, 30:1. So, green plants, what's their C-to-N ratio? So does this explain to you why you eat green plants? If your only diet is meat, you're going to be in trouble when it comes to way too much nitrogen, way too much phosphorus, way too much, and you're going to have to be pooping it out. And one way to settle your stomach is to get that green plant material in there. So we need that balance. Nematodes. When nematodes are growing, nematodes have an exoskeleton. So exo, meaning on the outside, skeleton, comprised mostly of chitin. So there's a cute word. Chitin, which is almost pure carbon. It's made of N-acetylglucosamine, so almost pure carbon. So this outside exoskeleton on the nematode is what makes the C-to-N ratio of that nematode so high. Carbon nitrogen ratio 100:1. Now yesterday we talked about the difference between green plant material and brown plant material. So we've already gone over that. Green has all those sugars, proteins, carbs. The plant pulls the sugars, proteins, carbs, stores them in the root system, so it's standing dead. Much higher in carbon, 150 to 200 carbons per every 1 nitrogen. Wood, C-to-N ratio, much wider. Conifer wood, much wider because of the tannins and the phenols, the terpenes that are typically in conifer wood. Why does conifer wood have so many of those terpenes and tannins and things that prevent microbial growth? To prevent microbial growth, and conifer is protected from fungi and bacteria trying to decompose it. Hardwood of cedar, C-to-N ratio 1,000:1. So you understand why we used to make our roofing material out of cedar shakes, because there's not many things on this planet that can decompose that material. So, we talked about this yesterday when we were talking about compost and the different kinds of materials. So I'm just going to zip through. Now, the first flash, high nitrogen, as it gets diluted by photosynthesis. Now we're at that normal 30 to 1. Flowers, 30 to 1, 10 to 1, somewhere up in that range. So we're taking nutrients out of the leaves and the shoots. Leaves and the shoots become much wider. So the end are flowers and seeds, 10 to 1 ratio on our seeds. And now after the seeds have left, the plant's going to take all those nutrients that are left, translocate them down into the root system, so the standing dead. And the difference between straw and hay. And now you all use that terminology correctly from now on, right? We're training everybody to use that terminology, right? So I think we got through this, and everyone's good with those conclusions. So bacteria and fungi are more concentrated than anything else on this planet for all of these nutrients. Do bacteria or fungi by themselves poop out nitrogen or poop out phosphorus or anything else? They can't, or they die. They are holding absolutely everything. So our nutrient retainers are the bacteria and fungi. The leachable forms of nitrogen are the soluble inorganic ones. Those are sigvaidal. What's the least leachable nitrogen? Yeah, bacteria and fungi. And then protozoa, because some of the protozoa hang out in the water solution and they will go with the water. Nematodes, some of them are hanging out in soil solution and they go when the water moves. So there is a gradient. If we really want to hold on to our nutrients, we must have bacteria and fungi in our soil. Do not let your nutrients end up in your groundwater, in your drinking water, in the ocean. We are destroying our ocean because we're allowing that kind of leaching. So, we have talked about that. Energy, where it comes from, how it gets down into your root system, that organic matter, who's using it, retaining nutrients in these 3 groups of organisms. These are our disease suppressors as well. Now, who releases the nutrients that are now held in our bacteria and fungi? We gotta have something that does that. And who are they? They're all these guys. So anytime we look at any of these arrows, nitrogen, phosphorus, sulfur, magnesium, calcium, sodium, potassium, blah, 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 is being released for your plants to take up. Most of this is occurring right in the root system. Why is that? Where's most of the bacteria and fungi food in soil? Coming from the roots. The exudates that the plants are producing are growing billions and billions of bacteria and fungi right in that area around the roots. So that's where all our nutrients are being retained. And now along come the protozoa nematode, because if you're a nematode, a fungal feeding nematode, where's most of the fungi in soil? In the root zone. If you're a protozoan, where are most of the bacteria in the soil? In the root zone. So we're going to concentrate all of this activity in the root zone. For as long as your plant is putting out the exudates, we are going to get this nutrient cycling revved up and going full gangbusters. When the plant doesn't need any nutrients, now what does it do? Yeah, it's been nice, guys. It's been really friendly. I'm glad you had a good party. But the exudates are being cut off now because I got to take my exudates and I'm going to make seeds out of them. Hi, see you next spring. And all that nutrient cycling slows down. So nematodes go someplace else. Bacteria and fungi go to sleep. They're going to be using whatever organic matter, but this whole nutrient cycle slows down when the plant doesn't need it. Isn't that nifty the way that works? So when we're looking at nutrient cycling, making nutrients available, precisely how does that work? We got to go back and look at C to N ratios. So flagellates require 30 carbons, 1 nitrogen. For every unit of biomass, a flagellate requires 30 carbons of 1 nitrogen. Protozoa eat bacteria, so that cute little flagellate is going to be eating a bacterium. Bacteria contain 5 carbons, 1 nitrogen. So see, we got all the nitrogen we need in our flagellate with just 1 bacterium, but look at this, we got a problem. Poor little flagellate doesn't have enough energy to go chasing another bacterium. Poor little baby's gonna die. Oh, wait a minute. What does he actually do? He goes and eats. How many more bacteria is he gonna eat?
5.
And so where he's hanging out? In the rhizome, because there's, all he does is have to turn around and, oh yeah, there they are, 5. So, flagellates are actually going to eat 6 bacteria. 6 times 5 is 30, so life is wonderful. Now that cute little flagellate is going to have all the energy he needs to go looking for more food. But he's got another problem. Isn't that amazing how nature works? Solve one problem, now you got another one. Way too much nitrogen for that poor little flagellate that's going to cause nitrogen toxicity. So what does that flagellate do with the excess nitrogen? Happens in soil. Protozoa poop is ammonium. What is released the most part out of any of our predators is going to be that excess nitrogen in the form of NH4. Is this plant available nitrogen? For perennials. It's plant available nitrogen for perennial plants, but it's not really helping your weeds any, is it? What has to happen to convert that into nitrate? What is the process? How does that happen? So all of a sudden we're going to have to look at nitrogen cycling. I just want to go through a few more calculations just in order to totally blow you away. So try not to get too lost. You can come back to this page over and over and over again if you really want to figure this out. A lot of people just gotta go like, I trust you, or you can do it yourself. When we look at nutrient cycling and these predator-prey interactions in our soils, do these predator-prey interactions supply enough plant-available nutrients or not? So let's go through those calculations. We have 5 nitrogens released for every 6 bacteria consumed. Each protozoan eats 10,000 bacteria a day. So we've got to have, what is it, 10,000 divided by 6? That's the units of biomass in a protozoan. So if each protozoan is eating 10,000 bacteria, they are releasing 8,000 molecules of nitrogen per day, per protozoan. A healthy soil contains 50,000 protozoa typically. Good, healthy soil. 50,000 protozoa per gram. And therefore, if we've got 50,000 protozoa, each of which eat 10,000 bacteria every day, our protozoa are eating 500 million bacteria per gram of soil. Our plant really needs to be pumping out those cakes and cookies, huh? To be growing that many bacteria. And if our protozoa are eating 500,000 bacteria a day, we are releasing 400 million molecules of nitrogen each day per gram of soil right around that root system. If you want to have some fun with Avogadro's number and all of that, convert this into the nanograms of nitrogen per surface area of your root. And so we are producing that number, 7 nanograms of nitrogen per area of your root system. So what is the nitrogen uptake of the fastest growing plant on this planet? This is Arabidopsis. Arabidopsis, the most rapidly growing plant on the planet, unless somebody knows another one. Then you can sit and watch Arabidopsis grow. It's got kudzu beet. How many of you have ever sat in a plastic chair at the edge of the road with your mint julep in hand and watch the kudzu grow across on the electrical line from one side of the road to another in about 2 hours. And Arabidopsis is faster than that. So of course, it's the lab rat plant. Because we want to get done with our scientific experiments really rapidly.
Where does that plant grow? I mean, in the
In the real world?
Yeah.
You know, I don't really know. In laboratories. It goes in labs. I would expect it's a tropical plant from someplace, but I've only grown into the laboratory. And I mostly take other people's word that this is the rate of uptake of nitrogen. 0.2 nanograms of nitrogen per root surface of the plant. So if our plants are taking up, and this is a max rate, if they're taking up 0.2 nanograms of nitrogen, but we are producing 7 nanograms of nitrogen, is the typical numbers of bacteria and protozoa, are we supplying enough nitrogen to grow any plant that you want to grow? Absolutely. We just have to have enough numbers of bacteria and fungi. We have to have enough food to keep those bacteria up and growing. What happens with the excess nitrogen? Because, you know, we're only taking up 0.2 nanograms. We got 7. So what happens to the other 6.8 nanograms of nitrogen per surface area of your root? Bacteria and fungi eat them. Your plant's already maximized what it needs. It's already taken up what it needs. So whatever's left over, the bacteria and fungi say, thanks plant, it's been fun. Now, I'll deliver some more tomorrow. So you keep your bacteria and fungi growing, you keep your plant happy. Now, we really only have data like this for nitrogen. We don't have data for the other micronutrients, but we're presuming it's the same. Do we maybe need to look at that? Yeah, maybe.
Does it make a difference between night and day? Like, when is the plant most actively growing?
It depends. It's the same answer even for me. Most plants will still be taking up nitrogen from the soil even during the evening in the dark hours. So you're a little beyond my knowledge of plant physiology. Sorry. So it's your charge to go find out. Nitrogen cycle. How do we convert ammonia into nitrate? What's the process? What's required? Always drives me crazy when I talk to chemists because they seem to think that this nitrogen cycle is a strictly chemical cycle. And nothing happens in the nitrogen cycle without biology. None of these conversions would happen if you didn't have these organisms in the soil. So we need to change that attitude of everybody. We need to change it into an understanding of the biology. So are you ready to take a voyage on the nitrogen cycle, or do you want to take a 10-minute break? Break? Okay, 10-minute break. Stand up, jump up and down.